Got a lot of terrific information to get through today, so let's get rolling here. Alyssa, we can go ahead and start to record. So hello, everyone, and welcome to the ATP-Bio public webinar series, which is co-promoted with the Society for Cryobiology to strengthen the cryobiology innovation ecosystem. I'm delighted to see so many participants in today's session entitled ATP-Bio, State of the Research. So my name is Tim Tripp, and I'm the Director of Strategic Partnerships and Innovation for ATP-Bio, and it's my pleasure to MC today's event. So we have a star-studded cast today in our session. So that includes 3 ATP-Bio researchers who are leaders in the field of cryobiology. So first up is Professor Korkut Ülgen. Dr. Ülgen is an Associate Professor of Surgery in Bioengineering at Harvard Medical School and Deputy Director of Research at Shriners Hospital for Children at Mass General Hospital. And in ATP-Bio, he's the Director of Research overseeing the research enterprise. Next is Professor Rebecca Sandlin. Dr. Sandlin is an Assistant Professor at Harvard Medical School, Mass General Hospital, where she is here today as the Thrust Area 2 leader in ATP-Bio. We'll also hear from Crystal Alvarez, who's a PhD student in Dr. Guillermo Aguilar's lab at Texas A&M University. And finally, we'll have Professor John Bischoff. Dr. Bischoff is a distinguished McKnight University professor in mechanical engineering and the Medtronic Bakken Endowed Chair for Engineering and Medicine at the University of Minnesota. And he's the PI and director of ATP-Bio, as well as the ATP-Bio Thrust Area 3 leader. So a big thank you to our speakers today for presenting the progress in the various areas of ATP-Bio. Before I turn it over to our speakers, I have just a few quick announcements on upcoming events for both ATP-Bio and the Society for Cryobiology. As I mentioned, this is a recurring webinar series, and we have 2 more sessions on the books for April 9th and June 4th. And coming up in March already, next month, is the Society for Cryobiology's webinar. In addition, the Society for Cryobiology has its annual meeting, Cryo 2024, coming up in July. Finally, ATP-Bio has other webinars open to ATP-Bio affiliated individuals, including the DCI Cafe, which is a terrific monthly listening and learning session about diversity and culture of inclusion ideas, and our ATP-Bio closed sessions that dive deep into research and other topic areas. So feel free to reach out directly to me or to others in ATP-Bio to learn how to potentially participate in those activities if you're not formally affiliated with ATP-Bio. And then finally, ATP-Bio is hosting its annual meeting at Texas A&M, our newest partner institution, in March. This is one of the highlights in the year for ATP-Bio and a great way to engage with the center. And likewise, please reach out to me to learn more if you're interested in attending that. So now it's my great pleasure to turn over the Zoom to our distinguished speakers, and we'll start with Dr. Ülgen, who will take us through the thrust areas and the research. Please, Dr. Ülgen, go ahead.
Thanks, Tim. Thank you for the introduction. You make this much more easy. Okay, it's great to have everybody here. Last time we checked, there were more than 150 people registered. And if you guys are like me, you register in the last 10 minutes because you realize there's no Zoom link you can access. So this is really wonderful. It's a very large audience. So I have 2 hats that I'm wearing today. One, as a director of research, I'll do like a very brief introduction of ATP-Bio. Many of you have already listened to this, especially at cryobiology, so I won't belabor. But if there are any questions, I'll be happy to answer. And then I'll drill down to, I'll basically present our first thrust area today because Dr. Stacy and Yarmish couldn't make it. And then I'll turn the floor to Becca and then to John. For questions and answers, this is a pretty loaded slide. So what we ask you to do is any questions you have at any time, just drop them in the chat. And once we stop the presentation, we'll start responding to those answers. We actually test drive this at the last NSF site visit, and it worked extremely well. Just makes better use of time. We'll probably have like 5 minutes for a more regular Q&A at the end, but it's limited time, and this felt like a more efficient way to do so. And also, you don't have to remember your question like for 40 minutes before you ask. Without further ado, we are very happy to present our updates on our research progress at ATP-Bio. This is intended as a highlights of research. There is simply too much content there to drill down scientifically into one thing. So instead, we'll basically give one slide highlight of everything we are up to. And this is intended to get you hopefully excited and collaborate or ask more questions. ATP-Bio is a National Science Foundation Gen 4 Engineering Research Center. These are the largest awards the engineering directorate makes. This is a national center. There are 6 major institutes, and then we have a bunch of collaborations nationally and internationally. Actually, there are about 20 plus, I forgot the exact number, 20 plus faculty and about 100 trainees and administrators and other staff at the center. This is a convergent research center, which means that this is not like everybody doing their own little thing and then publishing and calling it a center. We are actually tasked by the NSF to work very collaboratively, not just with other hard scientists. So it's not just engineers and scientists or biologists or doctors talking, but actually we collaborate very strongly with our colleagues across social sciences and law and ethics too. And there is actually a large number of publications and productivity and products coming out of that work as well. But given the time limit and the focus on society of cryobiology, we'll mostly limit our discussion today to the scientific bits. Our science can be basically categorized into 3 main buckets. One is healthcare, and here we do everything from cell therapies to organ transplantation to everything in between, and there's a lot of exciting work, as you'll see. The second one that we've been very active on is biodiversity, and this is everything from banking of species for research purposes to conservation efforts, especially coral reef cells, as you'll hear, as well as development of new vertebrate models, which is actually quite exciting to me. And then the third bucket is food supply and sustainability, which is somewhat new compared to the other ones. This is something more of a burgeoning field, and we are trying to develop these more strongly. So this is definitely one that we are looking into collaborations with, especially at the Society of Cryobiology. For collaborations, we have 4 main mechanisms. 2 are called, the first is RFP program, research program. This is basically a small grant application, 1 year. It needs to be in collaboration with an ATP-Bio faculty. We define the areas annually, areas of interest that is identified by research leadership, where we need either more expertise or new ideas or additional work done. The 4 major areas are on those bubbles. This will be announced on our website, atp-bio.org, probably in a week or 2. If you're interested, please contact either me or Catherine. We run the research program together and we'll be able to help you or connect you. Many of the people on this call already know many of us at ATP-Bio, so I don't think this is necessarily hard to ask, but if you need help, we are there for you. There is a translational version of this program. It's basically identical, but the difference is it needs to be much more translational or commercial or clinical translation. Tim and Carl are on that program, and there the focus areas are identified by our industry advisory board, and they review the applications to decide which one will work best. If you actually need to start collaborating first before you can write a grant application together, then we have 2 exchange programs. One is ATAP that's specifically for trainees. If they need training on something like we published and you want to see how a human liver perfusion is done, this is one of the ideas that could apply to you. And then there's a visiting scholar exchange program for that too. And again, the contact info is below both the website and the administrative email account that goes directly to Catherine. So contact us through either way, and we'll be happy to discuss more and explain what's being looked for. The research in ATP-Bio is split into 3 trust areas. This is now NSF jargon. This is how these things are designed. In trust area 1, we have biological engineering because our goal is to preserve living systems, biological systems. So we want to make sure that these, whatever the system is, can actually handle the exposure to CPA and the dramatic thermodynamic changes we are going to expose to them. And then when we bring them back up and get them ready for deployment, they'll be actually able to survive and function the way they're supposed to. Trust area 2 is a little bit more traditional cryobiology, where we focus on the thermodynamics of water in whatever the system. And there are many questions in terms of scale and application there, and Becca will present that. And then trust area 3, we make sure that the biological system is brought up to deployment temperature in a rapid and uniform rewarming. Many of you know this better than I do, the issues about ice risk, crystallization, and everything else. So there's really breakthrough work, and John will present them in TA3. So the trust areas encompass about 7 or 8 projects each. These we call core projects, and they are kind of like tied to our Gantt chart to make sure that we make progress the way we promised to our funding agency, National Science Foundation. But whatever new technology you are developing, then you apply it to one of our test beds. Now, in traditional engineering research centers, test beds are literally test rigs. They're not the real thing. They're just make sure that like the thing doesn't break immediately. It's different for us. Our test beds are actually products too. When we, let's say, vitrify a hepatocyte, a human hepatocyte, sure, that's a wonderful test bed. Our technology is working and everything, but we can literally provide that to a pharmaceutical partner so that they can test their drugs in this in better hepatocytes. And the same thing applies to across all test beds. We have 4 test beds. The first one is cells primarily intended for cell transplantation, red blood cells, iPSC and derivatives, engineered immune cells, as you all well know. CAR T cells are a complete revolution in immunology and cancer treatment these days. And then we have microphysiological systems. This is especially led by Kevin Healy at University of Berkeley, where we have these microfluidic systems that can recapitulate the organ processes in much more detail that we can identify. They also encompass things like islets or spheroids and organoids, which we are getting more and more into. Next, we have our organ test bed, and we are really proud how both horizontally and vertically integrated we are. We basically do work on any kind of solid organ, including vascular composite allografts, and also across all species, as you'll see in some of our presentations, everything from zebrafish to human donor organs we work with. We even have a couple of clinical trials. These what we call associated projects, because UNICEF doesn't fund clinical trials, but regardless, they're tightly integrated to ATP-Bio. And finally, we have whole organisms, and especially in biodiversity and sustainability and agriculture and aquaculture. These are key test beds that we use. This is my one real bragging slide, and I apologize, but I couldn't help myself. We presented to the National Science Foundation too. So Standard Engineering Research Center publishes about 25 papers per year. This last year, we are almost at triple that number. And if you dig into the detail, like who's publishing with who, it's not just like 2 or 3 very active centers publishing a bunch of papers and calling it a day. All the papers, more than half the papers have like either multiple institutions or have feature multiple disciplines. This is not just like chemical engineering and mechanical engineering either. It's like MDs, lawyers, ed degrees publishing jointly. And then we feature our trainees very well. I'm really proud of that. It's not just there on the papers. They are the lead papers, lead authors on the papers, but they're also on our patent applications and disclosures. So we try to support them and create the next leaders of the field. So I'm now taking off my administrative hat and putting on the scientist hat and starting our work on Trust Area 1. The faculty you see on the top row here, and the bottom line you see all the trainees. In usually our presentations, we put the pictures of the trainees. These are PhD students, technicians, postdoc, who actually did all the work, so you can actually, we can assign the credit. But as you'll see, the slides are so busy, we didn't think that would actually be more confusing. So here, again, I apologize, I can't do one by one, but this is all their work. We are faculty, we just brag about it. They're the ones in the lab that are actually doing all the work and making these really, really revolutionary technologies work. So biological engineering trust area, we start by thinking about the CPAs. Everybody knows the MSO and glycerol. We want to bring in new ideas. And it's not that sometimes we develop new CPAs, but it's not even that critical. We want to bring all the exciting new molecules that are identified as therapeutics in I don't know, the field of cancer is full of them because they want metabolic substitutes for treating cancer or reducing angiogenesis too. So we want to see if those things actually work for our purposes too. And then we want to make sure that we can get these CPAs into our system, cells or organs or whatever without creating injury. As you well know, sometimes they don't actually want to get into the cells or don't get to the mitochondria, and the delivery is always a problem. And then you want to make sure you're getting rid of these molecules because you don't necessarily want your DMSO, 1 molar DMSO going into a patient. So that's the last part of the problem, so that we have the system ready to deploy. And then finally, to make sure that the system is ready, we want to be able to diagnose the system so that the viability, quality, safety of the system is ready, especially if it's a clinical goal. So I'll go in that order, go from small to high basically, and end up with the QC. So I mentioned CPA. So this started as basically borrowing an existing pharmaceutical. It's called emricasan. It's a pan-caspase inhibitor, and it was tested in clinical trials for organ preservation. It didn't really work very well. It was like a marginal effect. So initial hypothesis was that, well, that was in static cold storage. What if we put this in the perfusion system? And it actually looked promising, but we didn't necessarily know why. So Başak and Brad actually, and Yoon actually took them down and tried these on cultured individual cells. And they found that endothelial cells are actually what these things are making clear impact. Now, Charles published this ages ago. We know that the endothelial cells are very susceptible to cold ischemia, but we show that, or rather, Başak and others show that this is through apoptosis, and the pan-caspase inhibitors actually work. And they actually leveraged another cocktail that includes emricasan but has 3 other additives into it. And then first tested it in vitro, and now took to ex vivo rat organs. And this paper is in preprint. You can find it already. It's going peer review right now, should be accepted soon. So here we have 19-minute warm ischemic rat livers. This is too much even for rat livers. So if you do a regular warm ischemic liver and then perfuse it, the resistance looks very bad. The resistance is an indication of endothelial injury, of course. And if you add our treatment, then suddenly it looks basically as good as a fresh organ. So again, we are taking this to other test beds, other trust areas, working with other investigators. There are these little boxes at the bottom called integration. They're there because we are a convergent research center. We want to make sure that we are not just working in our own bench doing our own thing, but working with others. So for instance, there are 2 papers. One is being reviewed, the other one is just being submitted to the American Journal of Transplantation on the impact of what would extended organ preservation do to organ transplantation, logistics and structure, and the ethics of organ sharing and everything. So this was one CPA, novel CPA, and that's great because thanks to ATP-Bio, because we are an integrated research center, we can play between human livers to rodent cells to rodent organs, and then go try it with hearts and everything else too. But we also want to be able to screen for these things efficiently, and this is really a brilliant idea of Shannon. She is the lead on this. So zebrafish are a very common model in biology. They are basically better than mice because you can very easily grow these things. You can very easily genetically modify these things. They are actually translucent, especially the embryo, so you can image the fish when you make a change too. They are usually used for developmental biology. You change a gene and see the jaw is not properly growing and that kind of thing for functional genetics. But turns out you can do transplantation and work with zebrafish too. Specifically, you can very easily take let's say a heart out and then try your CPA and it's actually beating on the dish. And then you see if you have a negative impact, some toxicity, etc. Turns out transplantation is very easy too. You just put the heart back into the fish and it just works. Don't ask me, well, I don't understand the biology, but I've seen it under the microscope. The heart pumps, things flow around and the fish swim. So they show that this actually can be used as a very high throughput model to screen for different CPAs in a whole organ system and actually an organism system too. And they are identifying cocktails to use for cryopreservation research. Next, this is led by one of our young investigators there in Savannah. So we have CPAs, but as you guys know, not all of them permeate into the cells very easily, and it's very tricky to actually deal with that. So they have this wonderful microfluidic technology where basically they run the cells through very narrow channels where there's shear, which actually elongates the cell. And along with the elongation, you're actually opening the pores within the membrane too. And what you see on the figures here is that they can actually put FITC labeled dextran into these cells that normally don't go in there very efficiently. And this doesn't really affect the viability or of the cell after this process, and they survive this very efficiently. Dextran is, of course, just the sample here. They've actually shown that this works with other cargo, especially CRISPR-Cas9 complexes, which is, of course, very exciting, but also with other CPAs as well. So this, we feel like, especially for cell transplantation test beds, this will be a very common and powerful technology. Going from cells to organs, so we are well known for our work in liver supercooling. One of the horizontal translation efforts is to take this to vascular composite allografts. This is face and hand and a number of other tissues. These are complex tissues. You have bone, you have skin, you have muscle, you have fat, and a whole bunch of other things. So they are much trickier to work. So we actually took first the supercooling idea to rat limbs, hind limbs. And the pictures you're seeing on the right are actually pig hind limbs. They are partial hind limbs, so we are basically chopping off the bottom and the top of the leg. So you only have the knee and therefore the bone and the cartilage and everything in between. And what we are showing here is that after, I believe this is 2 days time point, you can actually supercool. They don't freeze. They actually preserve much better in supercooling compared to static cold storage. And we've also shown that you can perfuse these hind limbs and transplant them successfully. That paper was published, I believe, last year, and the supercooling paper is in review and it should get published very soon. It doesn't work very well, though. The supercooling, we can't quite transplant yet. And Ken and Başak are on top of that. As I mentioned, they already identified the endothelial cells are the culprit. It seems doubly the problem in VCA, where the capillaries are even more prone to injury. So now they are working together. They actually have one of these RFPs that are jointly funded to identify how we can actually improve for these more complex tissues. So something that works quite well is partial freezing. This paper was published back in summer of 2022 for rat livers. We showed that we can freeze livers and tow them back in ex vivo in simulated transplant. They work quite well, up to 5 days. Within the last year, we've actually already scaled this up to pig and human livers. The video was for a pig liver that was actually frozen and about to get thawed. We actually improved this process. That's what the figures on the left are showing. And again, the endothelial cells were the culprit in this case. Just adding PEG and improving the protocol helped that. But this is working extremely well. We've already taken the pig livers to 10 days, and they look very good in simulated transplantation. We've already tried a few human livers, and they're not looking bad either. We're hoping this will be one of our next big papers. And on a horizontal translation, we actually tried this with pig kidneys too, and that is working extremely well. We've gone up to 30 days with frozen kidneys, and they are doing extremely well on the pump. So our next goal is to actually transplant them in a large animal model and see that if we can prove this for the first time in my knowledge anyway for sub-zero preserved organs. I talked about livers and limbs, a little bit about kidneys, but if you want to work with whole organs, pig, human, rat, you name it, lungs, hearts, muscle, vascular rings, bladder, uterus, Olizo is your man. The Visual Heart Labs in Minnesota is just awesome. If you haven't been there, please go and do it. It's just the most amazing thing ever. They do this for everything. They work very closely with with John and others on, especially the cryopreserved vitrified organs. And they just do it all show. It's like it's just a deal lab to go to. This is my last slide, and I'll stop. I mentioned quality assessment of these tissues and organs, the biological systems in general, to be ready to get them ready for deployment. So this is Shannon, and this is actually a collaboration with a company called Pandar, as well as with Paul and Tim in Minnesota. So here they're using something called resonant Raman spectroscopy. This is a twist on regular Raman, and basically there's this ratio which correlates very nicely to mitochondrial membrane potential, which itself, of course, correlates to the healthy state of the mitochondria. The figures on the top row, ADP, ATP, oxygen, and lactate, show you that up to 1 hour more ischemia, these levels are kind of okay. But if you extend this ischemia up to 3 hours, suddenly everything is bad. The ATP falls down and the ratio goes up. Oxygen consumption is less. They're very ischemic, you can see from lactate. And our 3-hour RMR signature, and this is a very easy, non-invasive, basically a 1 minute, you shine the laser, you read the reading, within 1 minute, you know. There's actually a large animal study of this, and we actually want to do a clinical trial using this approach too, which will hopefully open soon. But somewhat more new, and I wish Shannon was here to explain better than I do. But Raman's spectra is very rich. It's not just like it's good or bad. You can actually tell where you are getting the signature from, which means you can actually, within the ETC complex, you can identify certain points which are problematic. And they identify that the ROS generation between complex 3 and 4 is definitely an issue here. Now, it's not surprising that ROS is an issue, but the fact that you can get this from Raman signature is very novel. And we are thinking the same approach can actually be used to identify novel therapeutic targets as well. So with that, I'll stop. Thank you very much for listening and your time. I see a couple of questions on the chat. If they're to me, I'll answer them. But in the meantime, I'll give the floor to Becca to continue with Trust Area 2.
She sets that up. I'll just encourage folks to put their questions in the chat. Go ahead, Dr.
Everyone can see my screen and can hear me. I'm Rebecca Sandlin. I'm a faculty member at Mass General Hospital and Harvard Medical School, and I'm the Thrust Area 2 co-lead, along with Dr. Boris Rubinsky from UC Berkeley, where we're focused on the multi-scale thermodynamics of water. So work under this thrust is led by a number of researchers from academic institutions. I've highlighted both the faculty as well as the trainees associated with these projects. And if there's any questions you have that we can answer during this talk, feel free to reach out to a prospective faculty member. Their picture will be shown on each project slide. They'll be happy to discuss this with you. You can also reach out to me and I'm happy to connect you. Okay, so work in Thrust Area 2 is really focused on controlling water during the cooling process. So the whole purpose of cooling biological specimens, of course, is to suppress their metabolic rate as a way to extend their shelf life, where the lower we go in temperature, the more we suppress metabolic activity. So here we're primarily looking at 3 different storage regimes. This includes first high sub-zero ice-free preservation that we achieve through supercooling. So here we're going to sub-zero temperatures. We're getting the benefits of sub-zero temperatures that give reduced metabolic rate, but we're completely avoiding ice, which can of course damage our specimens. We then go to lower temperatures where we do have to deal with the issue of ice. And here we're looking at using partial freezing or isochoric cooling as a way to control both the timing and location of ice crystallization to minimize damage. And then we go all the way down to cryogenic temperatures for very long-term storage, where metabolic activity essentially stops. And our primary approach here is through vitrification or ice-free preservation. So regardless of the approach we're taking, each of these different storage regimes comes with its own unique set of barriers. And so ATP-Bio researchers are focused on overcoming these barriers as a way to preserve cells, tissues, organs, and even whole organisms. So the first project I want to highlight is work from Dr. Usta's lab at MGH in collaboration with Dr. Yarmish. And here they're looking at using supercooling to preserve specimens. So again, supercooling just means we're going to sub-zero temperatures, but we're avoiding ice. Now, the issue, of course, is this is a thermodynamically metastable state. So freezing probability is going to increase as a function of larger volumes, increased storage durations, and lower temperatures. So here the Usta lab is looking at ways to stabilize the supercold state and completely avoid ice nucleation. So here, the specific approach being taken, which was initially published in 2018, is what they refer to as surface sealing. So here, they're eliminating the air-water interface, which is a known site of ice nucleation. And in doing so, they did find in aqueous systems, they're able to maintain super cooled samples that are completely devoid of ice. So as part of ATP-Bio, they're working to translate this technology for the preservation of red blood cells. So RBCs are really interesting from a clinical perspective because of transfusion medicine. RBCs are being used in the clinic every single day. So in the United States, the RBCs are typically stored for 42 days at 4 degrees C refrigerated temperatures in a chemical solution. During that 42 days, the RBCs begin to degrade and eventually they have to be discarded. So what Dr. Usta's lab is looking to do is suppress that temperature lower. So right now they're stored under refrigeration, but if you can suppress that temperature lower to sub-zero, perhaps you can extend their shelf life. And his goal is to get those out to 100 days of storage using this super cold approach. So here, the data that I'm presenting from his group is where they took a series of RBC preservatives. These are commercially available preservatives, and they compared them to determine which ones are most compatible with super cold storage. And then they benchmarked that against refrigerated storage, which is the gold standard. After identifying the most successful storage solution, they then add in additional additives such as antioxidants to further stabilize the RBCs. And so now they've been able to demonstrate that going out to 161 days of storage, they're able to maintain a rate of hemolysis, which is RBC lysis, to below 5% compared to the refrigerated controls, which you see about 8% RBC lysis. So now they're working to further suppress this rate of RBC lysis to below 1% because that is the clinical standard currently used. So now we'll talk about a couple of projects in vitrification. So vitrification is where we're preserving specimens in an ice-free state. So here we're loading in high concentrations of cryoprotectants, and we're cooling them very quickly through the glass transition. So this prevents water molecules from coming together and actually nucleating ice. And so you're able to preserve specimens at cryogenic temperatures completely devoid of ice crystals. So this would be a glassy or amorphous solid. And this is a classic image I'm showing here from Dr. Greg Fahey's lab at 21st Century Medicine, where he took 2 rabbit kidneys, one frozen using slow cooling and the other vitrified. So visually, you can appreciate how useful vitrification is for multicellular systems such as tissues or organs. So in TA2, what we spend a lot of time doing is looking at how do we maximize cooling rates and minimize CPA concentrations. And this is because of the correlation where if you can achieve if you can achieve higher cooling and rewarming rates, then it requires less CPA concentration in order to vitrify. So the first project I'll highlight is our work to cryopreserve Cryptosporidium hominis oocysts. So Cryptosporidium is a parasitic organism that's responsible for really severe enteric illness. It's a leading cause of diarrheal illness in low and middle income countries. And there's really no effective drugs or vaccines. And a lot of that is because this is a very difficult organism to work with. There's no method to propagate the organism in vitro. And until now, there's been no way to cryopreserve it. So working with Cryptosporidium hominis requires you to propagate the organism through susceptible animals. And in the case of this specific species, that is a non-abiotic piglet model. So this is a very expensive model, and only a few labs are actually qualified to actually work with this animal model. So in collaboration with Dr. Saul Zapori at Tufts Veterinary School, he's a Cryptosporidium expert, we started looking at ways to cryopreserve this organism because it had never been done before. So here, the primary barrier was initially CPA loading. The parasite resides within an impermeable oocyst, and these are completely impermeable to water or CPAs. So after we developed a way to remove part of that waxy layer on the surface of the oocyst, we were able to show that we can load CPAs into the organism. And then we combined this with rapid cooling rates. And that's because we found that any ice crystallization whatsoever destroys the organism. So here we wanted to vitrify relatively high volumes of the parasite. So here we're going up to about 200 microliters. And initially we demonstrated that this is effective in microcapillaries, but because of the small volume, we scaled up to the vitrification cassette. And this is a device that we developed several years ago that has a relatively large footprint. So it's high aspect ratio, meaning that The length and width are quite large, but the inner chamber diameter is very small. It's only about 100 micron. And because we get such high surface area, we're able to achieve really fast cooling and rewarming rates. So combining those CPA loading protocols with the vitrification cassette, we were able to show successful vitrification of Cryptosporidium, where thawed parasites were inoculated into the piglet model, and infection did follow a similar course as the untreated controls. Another organism that's been really challenging to cryopreserve has been the mosquito. So first of all, why would we want to preserve mosquitoes? Well, they're responsible for the transmission of many diseases. So things like malaria, chikungunya, dengue, and these are really nasty infectious diseases. And so one of the vector control methods that's now emerging is genetic modification. So now we're at a point where we're seeing genetically modified mosquitoes released even in the United States. Now, the challenge with working with genetically modified mosquitoes is each strain you develop has to be maintained as a live colony, because there has been no way to preserve these. And so this is a challenge because it's expensive, it's time consuming, but you also have colonies that are susceptible to genetic drift or cross-contamination, which can destroy a respective colony that's been well characterized. So reproducible cryopreservation methods are really needed in this field. So here we've been working in collaboration with the CDC and the USDA to develop methods to first find low toxicity cryoprotectant cocktails, which we've shown here. And now more recently, we've begun collaborating with Dr. Rabin from Carnegie Mellon to develop thermal models to understand heat transfer throughout this organism. So having that information, knowing what our relative heat transfer is, and therefore the concentration of CPAs that we would need for vitrification, that can help guide further optimization of low toxicity CPA cocktails. So now I'll switch gears a bit and talk about some of the work in isochoric preservation. So this is really being pioneered by Dr. Rubinski from UC Berkeley. And here, the systems I've talked about to this point have been isobaric systems. They're open to the environment, and the system will freeze at equilibrium when being slowly cooled. So the isochoric system differs in that it's constant volume. So by restricting the expansion of the solution, you're able to reach a two-phase liquid ice equilibrium at sub-zero temperatures. So over the past few years, Dr. Rubinski has been working with Dr. Matt Powell-Palm to first of all, develop a range of different technologies to characterize the isochoric system. So that includes the recent development of the isochoric cryomicroscope stage, and they've also developed an isochoric nucleation detection device. But importantly, they also work with other investigators to translate these technologies. So for example, he's worked closely with Dr. Healy, also at UC Berkeley, to develop this isochoric supercooling approach for microphysiological systems. And then also the team has worked with Dr. Mary Hagedorn at the Smithsonian to apply isochoric vitrification to preserve coral reef fragments. Further work in Dr. Rubinski's lab has been with 3D cryoprinting. So the specific project I'm going to highlight here is work that they've done for cryoprinting food products for dysphagia patients. So dysphagia is a medical condition where the patients have difficulty swallowing, and this can lead to risk of malnutrition or death due to choking. And right now, the market standard for dysphagia patients is pureed food products. So these can be somewhat unappetizing and unappealing just because texturally there's not much there. So Dr. Rubinski has been working to develop this 3D cryoprinting combined with directional freezing to show that they can control both the macrostructure and microstructure of food products. And here, this is food that's been produced from a beef slurry. And the resulting food product, they did show that it has reduced adhesiveness, which minimizes the risk for choking, but it had improved textural properties. So it has more of a food-like mouthfeel that you would expect. The final project that I'll highlight is work from Dr. Chris Dame's lab, also at UC Berkeley. So here they're working to develop a 3-omega sensor to characterize the cooling and rewarming process of biological specimens. So the 3-omega approach relies on differences in thermal conductivities between crystalline and amorphous states. So you can see here this has been applied to water CPA model systems. They then translate this to tissues. And one of the issues that they found is whenever they're trying to characterize this cooling and rewarming process, they often would see a lot of condensation or frost form on the surface of the material, which interferes with data acquisition. So recently, they've developed this new probe design. It's essentially a needle design, and it can be inserted directly into the biospecimen to monitor ice crystallization during cooling and rewarming to help understand the difference between vitreous and crystalline states of water. So with that, I'm going to wrap up, and we'll have time for questions, hopefully at the end, but feel free to pop questions in the chat window, and I'll be happy to answer those.
Thanks, Becca. So I'll wrap up with thrust area 3, and Cristal from Guillermo's lab is also going to give me a hand here. You can see both the faculty as well as the trainees that participated, and there's at least one trainee missing, and that'll come up in my slides. So what we're trying to do in TA3 is really build on what's been done in TA1 and TA2. So we're preparing the biological system to go into suspended animation in TA1. We then take them into suspended animation in TA2, which Becca just presented nicely. And then in TA3, we're really focused on bringing them back. In many cases, this ends up being a case of needing rapid and uniform rewarming, and that's to avoid ice and cracking are two barriers, and there are a few more. So I'm going to start here by letting Cristal present what's going on in Guillermo's lab. Cristal?
Yes, thank you, John. Hi, everyone, I'm Cristal. So here at Texas A&M University and Dr. Guillermo Aguilar's lab, we are developing optical tools for in situ thermodynamic studies in cryopreservation. And so ideally, these tools that we are developing are digital holography and Raman spectroscopy with the goal of measuring thermodynamic information while we are cooling using various technologies such as supercooling or vitrification and also laser rewarming. And we are developing these tools with the hopes of understanding what is physically happening during this rapid changing phenomenon, and how we can use this information to enhance cryopreservation protocols and overall increase the viability of biological samples of various sizes. So digital holography is a non-invasive optical tool where we pass light through a liquid or transparent sample that is changing in temperature, and we are able to observe the changes in refractive index. And so through this refractive index information, we're able to reconstruct 2D temperature maps as the time is changing, and therefore we're able to measure thermodynamic information and also phase changes. So at the moment, we're able to detect when ice forms within the whole solution and also in certain areas, which is important for the field of cryo. And overall, this tool has a high spatial and temporal resolution, which is important for detecting these cooling and warming rates at various locations and also seeing if the temperature is changing uniformly. And this can be adjusted for various sizes, from droplet scale to milliliter scale. And at the moment, we're measuring temperature in large samples, as you can see in those figures there, in collaboration with Matt Powell-Palm here at Texas A&M. But we are also moving into rescaling this digital holography temperature measuring system to droplets to continue our collaboration with John Bischoff and also measure the temperature in droplets hopefully while we're vitrifying them and also doing laser rewarming to enhance the viability of samples. And in the bottom corner, that is our Raman thermometry system, which is being developed by my colleague Carla Berospe. And she's developing this Raman system to measure CPA penetration in biological systems. And she's currently measuring Raman in Artemia cysts that are loaded with different CPAs to see how far in the CPAs penetrate through the cyst. And she's also able to measure the Raman intensity in situ, so while she is cooling the cyst, and to see how that CPA is changing as the temperature is changing. And so overall with this tool, she can look at CPA in different depths and also observe the Raman peak shifts as the temperature is changing. And she hopes to scale this up to other test beds such as mosquito larvae in collaboration with Rebecca Sandlin at MGH, and hopefully translate it to other test beds as well. So overall here at A&M, we're combining both of these techniques in the future to have a full characterization in situ while cooling and warming to find ideal parameters to enhance the viability of various biological samples ranging from droplets to the milliliter scale.
Great. Thank you, Crystal. And we're all looking forward to visiting with you and Guillermo and Matt and others for the annual meeting. And I'll just point out in the right-hand bottom, the integration, which we probably won't have time to go through in each slide, but you can see there how the pillars are also overlapping. So thanks again, Crystal.
Thank you.
So moving on then to some of the work coming out of Beth Stadler's lab. She's focused on nanobars or nanowarming, and you can see that she's actually able to get a tremendous amount of heat out of these nanobars. They are currently not made of metals that would be used in the body, but this is a demonstration project, and they're far above 5,000 watts per gram, which is a typical ceiling threshold for iron oxide nanoparticles, which we do use. And you can see that there are different opportunities there in terms of alignment, in terms of different metals and other dials that can be changed in the material science world to get higher heating. She's collaborating with Rhonda Franklin, and they're also looking at using these nanobars for unique signatures that they can get actually. And you can see their IEEE publication there in the upper right. This would actually be something that would allow you to do something like an RFID tag on a nano or a biological system that was impregnated with these nanobars. Rhonda is also working, Rhonda Franklin's lab is working on RF characterization of CPA cocktails for nanowarming. And what I've shown there in the upper left is a coil, an RF coil with a nanoparticle impregnated liver in it. And the radius, as the radius goes larger, as you're moving from rodent systems out to human scale systems, you begin to experience higher and higher eddy current heating, which is basically, it goes with the radius directly. So we need to understand how much of an issue is that going to be. And Rhonda is an expert in microwaves and dielectric loss measurements. And so she has built with her trainees a coaxial probe and a split ring sensor that allows us to do characterization in different frequency regimes and in different temperature regimes, as you can see. So the frequency dependence is shown there in the upper left. That's in various glycerol solutions from the literature. And then you can see our experiments are overlapping with that, which is good. That's sort of a check. But then if you drop down from there, we can actually look and see that different CPA component losses are different. So we will be getting different eddy current heating depending on what our CPA components are. Up in our right hand, you can see the temperature dependence as you're dropping to lower temperatures, and that's a benefit of the split ring sensor. You can see that there is a very dramatic temperature dependence to this effect. And also there's an effect based on the concentration, as you see in the lower right. So all of these things are extremely helpful and needed for us to actually design RF nanowarming and other types of electromagnetic warming in the future. In terms of rapid cooling and rewarming, the cryomesh system continues to be one that we're investigating here in Minnesota and increasingly through collaborations with other groups. The latest work comes from Zhongqi Guo, and he published that in Advanced Science this year, showing that he can actually cryopreserve multiple different systems of different sizes all the way up to a zebrafish embryo in a high throughput scalable manner, starting with coral, moving through Drosophila embryos into zebrafish. This is really exciting because I had never seen anybody do anything as big as a zebrafish embryo on anything other than a cryotop, and that means one at a time. But now we're able to do that in a high throughput manner. You can also see the Joule heating work that came also from Lee's work, who's now at MGH. And this is a scalable technology where you're using a voltage pulse through, for instance, a mesh, a metallic mesh. And the Joule heating basically ends up being the warming that you need for the system. So there are certain limitations associated with this because it's a boundary warming application. And so that is actually what Zhongqi is now following up to find exactly where are the limits of this. How much can we do from an adherent cell all the way up to maybe even tissue slices and beyond. 3D printing is coming out of the McAlpine group, and this is being used for high throughput cryopreservation. There's both a patent pending and a paper that is out in review for this, and it's basically showing the ability. You can see his very impressive on the left upper corner printing setup, and he's able to print a variety of living biological systems, which is featured in the paper. And the patent relevant to cryopreservation is that we can actually manipulate zebrafish embryos in the upper right, as well as whole droplets full of, say, shrimp larvae. And then we can also use pick and print and other printing technologies to move, say, dead or non-interesting aspects of what's coming out of the cryopreservation. Maybe it's a dead embryo. You can remove them, and then you can basically clean up your sample. You can also print onto conformal surfaces. All of these things make it a very interesting and useful approach. There are novel nanoparticles coming out of the Riverside Mengolini group. You can see his trainees, Giulio and Aishwarya. And they are working primarily with titanium nitride, which is low cost and abundant. You can actually use it for in situ thermometry. So you can see that they can actually compare between gold alone, titanium nitride, and also hybrid nanoparticles that they're working on in collaboration with Yadong Yin's group there at UC Riverside. You also can use, he is using nanoscale Raman thermometry. And all of this is leading to new novel nanoparticles that can be used in the rewarming approach. Yadong is quite active in, as you saw, both his collaboration around laser nanowarming, but also for RF. And this is an example using a type of a resin, FR, which then leads to colloidal stability of these iron oxide clusters you see here. And the viability, they're basically very biocompatible, all the way up to 10 milligrams iron per ml. You can see some of the heating data that he's able to achieve with this, which is quite good. And he can go all the way up to 10 milligrams iron per ml and get heating rates that are quite reasonable and in line with what we might be needing to heat some of our larger systems. So this is an excellent example of colloidal stability that he can achieve in a very highly concentrated VS55 CPA, which is an ongoing struggle for all of us working in that area. In addition, Yadong is also working with using what he's calling stage-wise regulation of nanowarming, and he's using magnetic disturbances, so external magnetic fields to actually align magnetic nanorods in the field, and that changes, and you can see that in the slopes between the lower temperatures going into the higher temperatures. So by changing the magnetic alignment, he can sort of reduce the rates in different zones where we might want to avoid cracking or where we want to avoid overheating, as an example. So the vertical pre-alignment enhances the initial heating, and then the additional magnetic field disturbs it, and then that makes the heating drop, which I think is it's a very interesting approach, and there's a paper that is in revision on that. And in addition, Yoed's group, in collaboration, I think, with Dr. Kara, has been working on a number of approaches to prevent structural damage during preservation and rewarming. And you can see a few of those examples here in terms of preservation of blood vessels, liver perfusion modeling for CPA, which I think overlaps with TA1, as well as ovary preservation projects. There's also work coming out of Mike Garwood's lab, and here I did feature Soren, even though we didn't show all of the wonderful trainees that are doing most of all this work out of all these labs. But Soren just had one of his, I think his first paper come out in ACS sensors on this really interesting LOD EPR system, which is a way to very cheaply assess how much iron we have within a loaded organ or biosystem as compared to, say, NMR relaxometry. And this is really super idea, and I think it's quite scalable, and we're excited to explore what we can do next in this area. This work comes from my collaboration with Eric Finger, and what we're doing is trying to figure out how to load with a vitrification solution, and I think everyone has probably seen these slides before, but this is from some of our successful work with the rat kidney. And what's interesting here, of course, we're building on the excellent work of Greg Fahey, but we're able to get to something close to 92 to 95% equilibration of VMP in the tissue. But what was really something that I learned from all this is that we never really get 100% equilibration, but we can get up to something like 7.76 or 7.8 molar. And that, based on micro CT in our hands, is good enough to actually vitrify. If we put iron oxide in with that VMP, we were then able to show that we can actually do nanowarming. We can actually cryopreserve by vitrification these optimally loaded kidneys, and we can store them for up to 100 days, bring them out of storage, put them through the RF nanowarming, and then put them into an nephrectomized animal, and the animal survives up to 30 days. There is some initial 2 weeks or so of some of the blood markers going high, but then things seem to stabilize for the remaining days of the 30-day time frame of the experiment. So this was a really big win, and of course Zheng Hu as well as Joe Rao shared the co-first authorship on that, and it was picked up and came on to the cover of Science, and we were all very, very excited about that. We had both Susan and Tim also participate on that publication. So with that, I'll end, and I think we have just a few minutes in case anybody has any questions.
In the chat, one did come in for Dr. Sandlin. She seemed to answer it in there, but I'll just maybe rephrase the question so others can participate in that. So the question was around using your process for crypto in other microorganisms or infectious disease models.
Yeah, Tim, so that's a great question. The overall approach should be valid, but I suspect the biggest issue would be in CPA optimization, because we would expect there likely to be a different CPA cocktail or CPA loading protocol that would be necessary.
I suppose in treatment of the spores or the biological material may need to be different as well, depending on the coats. Good. Thank you for that.
In the absence of a question, I just wanted to point everyone to the chat. I put Mary's PBS little video there, if anyone's interested, talking about trying to cryopreserve coral. And also there's some other groups that are part of that, but this features some of Matt's work and Boris's work with her.
Thank you, John. If there are other questions, you feel free to come off mute and shout them out in the last minute here. I've got a quick one for Corkett. You know, thinking about the complexity of in tissues, even broader than a whole organ, essentially. How are you thinking about, or how are you working around the cryoprotectants really infusing into these various areas of a hind limb? Certainly there's different tissues in there. There'll be different permeabilities of these cryoprotectants to get there. Are you concerned about that? Is that something that you're dealing with?
I'm very concerned, and I have to tell you, I have no idea. I mean, when we do a VCA preservation, there is the whole bone in there, and we've never dared look what's going actually into the bone. But we have some new funding to specifically look there, so we'll hopefully learn a little bit more. But that will be a wonderful area for collaboration. I'm actually trying to follow what Becca and Purva are doing with their also use of Raman to identify where the CPA has gone. And I mean, it's not necessarily going to work with the whole organ, but at least I think it will inform us where these things diffuse or get delivered to, and we'll learn more about that.
Terrific. Thank you, Corgan. All right. Well, that brings us to 4 p.m. Central Time, 5 p.m. East Coast. Thank you, everyone, for participating in this co-promoted session, and it's been a great to hear from our ATP-Bio leaders. So thank you all, and enjoy the rest of your week.
Thanks.
Thank you all for being here and listening.